Good afternoon, everyone. This is Lynn Katz-Cherry of the Great Lakes Green Chemistry Network, and I want to welcome you to our webinar today on behalf of the network and the Michigan Green Chemistry Clearinghouse. We're very happy to have Jen Sass here today, the senior scientist of the National Resources Defense Council, who will be talking about the utility of the Greenscreen for safer chemicals for nanoscale hazard assessment. Well, today we have a terrific presenter, excuse me, Jennifer Sass, the senior scientist of the Natural Resources Defense Council. And I've had the privilege of knowing Jennifer for some time now, and I'm always really impressed by her depth of knowledge and her experience in the field of environmental health science. She has been the senior scientist at NRDC's Health and Environment Program since 2001, and she is also a professorial lecturer at George Washington University. She reviews the science underpinning the regulation of toxic chemicals and advocates for health protective regulations consistent with environmental laws. She holds a doctoral degree in cell biology from the University of Saskatchewan, Canada, and a postdoctoral certificate in environmental toxicology from the University of Maryland. And today she's going to do a presentation on a green screen that was done for nanoscale hazard assessment on a nanosilver case study. So I'm going to turn it over to Jennifer. Thanks, Jennifer.
Thanks very much for that introduction, and also for the invitation to present today. I'm really excited. I'm going to present to you a project that's still ongoing. So at this point, it's in the draft phase in terms of our final output, but I think that the project was a success in the sense that we were able to show that we could use Greenscreen to do a hazard screen of a nanomaterial, and that's what I'll talk with you about today. Our goal in using the Greenscreen is to identify safer ingredients, drive more transparency, and also develop hazard assessments, like a hazard level screening assessment. The reason why we focused on nanomaterials is because on all across the world, really, Europe, the US, and Asia, there's been a real resistance to testing nanomaterials through traditional toxicology testing schemes. The dialogue that's been happening is all around whether we need new testing schemes, whether these nanomaterials are so unique, whether their properties are so unusual, their properties are more changeable, that we need new toxicology to go with testing them. So I wanted to cut through that and say, no, even though these chemicals are unique and interesting and changeable in many ways, toxicology shouldn't change. We still want to know the same kinds of things. We want to know, can it penetrate membranes? Can it penetrate the blood-brain barrier? Does it cause cell death? Could it cause cancer? Could it cause reproductive changes? If inhaled, can it get from the lungs into the bloodstream? So all those basic questions haven't changed, and that's what this project was about. It was about cutting through that conversation and seeing whether we could do a basic hazard assessment. It was also about making the point that in doing a basic hazard assessment, we don't know very much about these materials, so they're inadequately tested. The reason why that's important is because the one we chose, nanosilver, has been approved by US EPA for use in textiles, clothing, blankets, sheets, pillowcases, all sorts of different textiles, including clothing for children. So we wanted to show not only the data that we did have, but also identify data gaps. And Greenscreen could do that for us. This slide has a quote from Dr. Linda Birnbaum, who's the director of the National Institute for Environmental Health Sciences and the National Toxicology Program, and she says, "We currently know very little about nanoscale materials' effects on human health and the environment. The same properties that make them so potentially beneficial in drug delivery and product development are some of the same reasons we need to be cautious about their presence in the environment." The nanomaterials are interesting because they are smaller and they're the same chemicals off of your periodic table, but they're designed and engineered to be much smaller, so small in fact that their properties change, and that's what's interesting about them for product developers as well as in medical uses. So for example, where gold is yellow, nano gold is red. That's interesting and entertaining, but maybe not useful. But there are other nanomaterials that when they get small, they become conductors if they weren't, or maybe they stop being conductors if they were at larger scale. They can pass through biological membranes easier, and the properties, they become maybe stronger. For example, carbon nanotubes are 100 times stronger and 6 times lighter than than steel. So their carbon nanotubes are used in a lot of products where they need to be reinforced much stronger. So the same kinds of properties that make them interesting for product development may also alter their toxicology. So carbon nanotubes look a lot like asbestos fibers, whereas carbon at a normal scale doesn't look anything like an asbestos fiber. So carbon nanotubes also act like asbestos fibers in our body. So these are the kinds of things that toxicologists were grappling with when they were trying to figure out how to test and ultimately regulate these materials. So again, the goal was to test whether the Greenscreen could do a screening level hazard assessment, and I'll talk more about the Greenscreen. But all we wanted it to do is take the data that's out there, organize it according to toxicological endpoints that are of interest to us, and identify data gaps. So again, I'll talk more about the Greenscreen. The approach was to convene a prominent group of independent experts, so we did from academia around the country and also in Europe, to discuss all of the categories of the Greenscreen, all of the toxicological endpoints, and try and figure out what is a nanomaterial that was of interest to consumers, that there would be widespread exposure, and that there was enough data that you could do a meaningful hazard assessment. If there was no data, it wouldn't be very interesting because you would just get a lot of data gaps. So we wanted something there was data. We picked nanosilver because there was a whole docket of studies, in fact, because EPA had approved it for textiles. We also picked nanosilver because of its use in textiles, we knew there would be direct human exposure, because of going into clothing and household items. Then we went through all of the endpoints of a Greenscreen, which I'll show you in a minute, and the scientific team helped us to organize the data. So we said, all right, what are the properties of nanosilver that are different than silver? Some of the things are size distribution. The silver tends to be a similarly sized molecule. If you have a whole bunch of silver, your atoms are going to be all the same size. Whereas nanosilver, the distribution ranged from nanoscale, so very, very small, up until larger. Some kind of range of distribution. It might range from a few nanometers in diameter up to several hundred or even 1,000 nanometers in diameter in a single sample of nanosilver. So size distribution was more variable. Shape may be more variable. Structure, charge could be more variable. If you coat the nanomaterial versus not coated, it may have very different properties. The surface chemistry is very important for nanomaterials. The surface is how chemicals interact with each other. And one of the things that makes nanomaterials so interesting is that they have a very small surface size per mass. In fact, their mass is almost nothing. And so their surface size, which allows them to interact with other chemicals, means that they're much more reactive per mass of the material than a normal size nanomaterial. They may agglomerate. It may be that all of your nanomaterials clump together and you don't actually have a nanoscale material anymore. That could be important, or it could stay dispersed. And it may do that in different media. So that's important to know. So these were some of the differences between a nanoscale material and a normal scale material in looking at data and doing a green screen. And so the scientific team recommended modifications to the green screen method, and we'll show you some of those. And then what we did was we started looking for an independent contractor. In order to conduct a green screen, a contractor has to be listed, has to be a certified profiler for the green screen. So Greenscreen does trainings, they teach people how to use it, and they certify different contractors to do, as profilers, to do the green screen. So we went to several different contractors until we found one that would take this project on. And then we had them meet with the scientific team multiple times to talk through the data available and some of the approaches. So we had a team that knew Greenscreen well, and we had a team that knew these nanomaterials well, and then we started the conversation back and forth. So what is a green screen? The green screen is a method. We chose it because it's been validated. It's been used broadly for comparative chemical hazard assessments. I want to emphasize that it's hazard only. It does not take into account risk. So exposure is not a parameter of it. It's just assessing how hazardous the material is based on the data available. It was developed by a nonprofit called Clean Production Action, and it allows one to compare chemicals based on hazard using a level playing field. So they all go into this validated method. It sorts the data that's available by different endpoints. It scores it overall based on what kind of a hazard score it gets, which I'll show you in a minute, and it identifies data gaps, which is important. It relies on the Design for the Environment approach of US EPA, and US EPA Design for the Environment uses Greenscreen regularly. It's also used by other companies that use it. Hewlett-Packard uses it regularly. IBM uses it. States have used it as well. It's built into some of the California green chemistry and safer consumer product regulations. So it's a very validated, well-used sort of standard method. And so we knew that although we were altering it in some ways for this nanomaterial, for nanosilver, we knew that in general the method would be accepted, and we would have to identify where we'd altered it. Ours considers 18 environmental and human health endpoints. That's more than a standard green screen. Oh, sorry, we added some to that. The standard is 18, and I'll show you those. And it addresses also breakdown products. So in other words, if a chemical, for instance, a polymer, for example, a plastic, for example, might be non-hazardous when it's polymerized, but if it breaks down into hazardous materials, then it would get a hazard score because it is a life cycle assessment, and it's hazard only, not exposure, and it does consider the breakdown products. And then it gives it a final score called its benchmark score. It's used, as I said, by a number of different corporations, including Hewlett-Packard, Staples. Some of the states have used it in their regulations. US GBC, Green Building Council, uses it in their LEED standards that are proposed. So when companies score chemicals for hazards to be used in buildings, LEED accepts that they use the green screen to score those and choose a lower score or a lower hazard chemical according to the green screen. And it's also used for alternatives assessments, such as I suggested the California Safe Consumer Product Act. So it's quite well accepted as a method on its own. These are the 18 different endpoints. So there are some human health ones that we care about: carcinogenicity, mutagenicity, reproductive tox, developmental, and endocrine activity. This is not completely comprehensive, but I think it captures what we want to know at a screening level. It has also some acute toxicity, systemic toxicity, neurotoxicity, skin and lung sensitization, and skin and eye irritation endpoints. Again, if there's data, it would fill into these, and if there's not data, it would be identified as a data gap. if there is data, it would be scored or ranked on a lower or higher hazard. There's some environmental toxicity and fate endpoints, because as I pointed out, it's a full life cycle hazard assessment. So it considers acute toxicity, and again, it's not about exposure. The material may be used in ways that never gets into water, but here you would just point out if it was hazardous to aquatic organisms, acute and chronic, and other ecotoxicity where available. And also some of the characteristics we care about, like persistence, bioaccumulation, and then physical hazards like its chemical reactivity and flammability. So there's some things about nanomaterials that are kind of different here. Like, for instance, the nanomaterials tend to be very persistent because they tend to be, kind of atomic. They're sort of at your basic level of your of chemistry, so they can't be broken down further. So they tend to persist in that state. It's not true for all nanomaterials, but it tends to be. There is some suggestion that they could be flammable, and that's because, or explosive, and that's because they are like a dust. They're so small that there's a suggestion that they could act like dust. And OSHA, in fact, applies the dust standard to some nanomaterials because of that concern. And that, again, that's just based on their very, very small size. So there's some things about nanomaterials that are different that we consider. We assign a level of concern for each endpoint based on high, medium, or low. And no questions on that, so I will move forward. The idea is to help people make informed decisions. People could be individuals, they could be NGOs, they could be companies developing products in the R&D phase or later, and they could be governments. A lot of times it's to advise companies about making safer choices. It points out, as I said, what you know and what you don't know by identifying data gaps. It has different benchmark scores. It will give you an overall score of 1 to 4. 4 is an inherently low, and 1 is high hazard. It's to avoid or phase out. So the idea is to help companies choose more of the lower hazard ones and avoid the higher hazard ones. So for instance, a LEED standard might only give a credit if the company is not using any chemicals that fall into the benchmark 1 or 2, for example. I made that up, but it would be that kind of thing. It can be used also by non-experts in toxicology. So nanosilver, what is nanosilver? There's ionic silver, which are these charged ions like Ag+. There's particulates, nanoparticulates, and then there's the bulk silver. So there's pictures of each one. The bulk silver would be like your silver spoon. All of them release ions, release these Ag+ ions, and it's the ions that is the toxicity. It's the ions that damages cells and causes toxicity. It's the ions that also makes it effective. So it's used as an antimicrobial because it kills microbes. It kills germs. It kills bacteria. Silver is registered as an antimicrobial already by EPA and has been for years. Nanosilver is considered to be even more effective than silver, and that's because it releases more ions off its surface area per mass of material. You can have a smaller amount of nanosilver than silver and get more ions released, which is more effective antimicrobial. Because of that use, it's used in food packaging, food cutting boards, clothing, fabrics. It's also used in some medical applications like wound dressing and infection treatments. And as well, it could be used in washing. It's used in some washing machines and paints. These are a real problem. I'm really concerned about these uses. There's some washing machines you can buy that release nanosilver ions into every load with the idea that it's sterilizing your clothing. Those are really concerning to me because, first of all, nobody needs sterile clothing, and second of all, that's a direct release into what will be wastewater. The wastewater treatment folks are very concerned about nanosilver being used because it is likely to impair or kill the beneficial bacteria that they need to treat sewer. So bacteria are required for treating sewage, and if there's too much nanosilver in the wastewater system, it could destroy that system. So there's been a number of letters written to EPA by wastewater treatment folks that are concerned about this use. Okay. So we looked at 3 different kinds: nanoscale metallic silver, the one that we're really concerned about, a silver silica composite. This is the one that was actually approved by EPA. It's called AGS-20, made by a company, approved for textiles, and then conventional silver, like a silver spoon. Actually, we used dispersed silver because silver in a glass of water, basically, because we thought that was the closest analogy to nanoscale and the composite. The extent of the nanoscale test material characterization was considered. So if we had no data on it, then we called it a data gap. And so here is what we found. I'm listing these as draft results for right now because the contractor, we're having these peer-reviewed, and we still have some questions about some of these. So these are draft for right now. But the very top Greenscreen is regular silver. It got a benchmark score of 1 overall. It's very high toxicity, and that's because it's an antimicrobial. It's, like I say, it's registered as an antimicrobial. And again, this is not your silver spoon. This is dispersed silver, normal scale silver, like in a glass of water. It's very, very highly toxic to aquatic organisms. In fact, silver is more toxic than mercury to aquatic organisms. It's also persistent. It got a red mark there, very high under the P. And it's got a lot of data gaps, but otherwise where there is data, it's fairly low toxicity to humans, but very high toxicity for acute and chronic aquatic toxicity. That's the AA and the CA. And the P column under fate for persistence. And that makes sense. We know about that with silver, so that's not a surprise. The next chart is for nanosilver. This is the ones that release ions more. It also got a score of 1. It was based also on the same as silver, very high persistence. That's the red for the P column, and very high aquatic and chronic, acute and chronic aquatic toxicity for the ecotoxicity. In addition, there was a high concern from some of the toxicity studies, both cell and animal ones. Acute toxicity, particularly, it got a red very high in the inhalation. There was concern that the nanosilver could be inhaled, whereas the regular silver isn't. The nanosilver is so much smaller that once inhaled, it could get into the bloodstream, and there was some toxicity associated with that. So that was a concern. At the bottom, we did the AGS-20, which is the silver silica composite that was approved and by EPA. And what you'll notice here is it's very high for persistence, and it actually has a medium for eye irritation. There was some concern there. But actually, there was a lot of data gaps, and that's a real concern for us. Mostly all of those DGs sprinkled all the way through this chart are data gaps. So for us, that was a concern. Now, EPA, when they approved it, they substituted other nanosilver and other silver data to substitute for those data gaps, but our scientists said that we should probably not substitute. And in fact, EPA's own advisory committee said you shouldn't substitute because we don't know enough about these materials to know whether we can substitute one kind of nanosilver for another kind or silver for nanosilver. We just don't know enough about it. So we decided not to substitute, and because of that, we had a lot of data gaps. But again, this is still draft. This is just a wording summary of what I found, what we found. This I don't want to go into too much, but it basically says there's a number of different methods for incorporating silver into fabrics, that not all products are alike, and that's something to consider. that some it may come out of more and some it may be bound into more. For example, some companies sell clothing. Like I know there's yoga and workout clothing that has nanosilver in it. They claim that the silver is in these threads and so it doesn't come out of the material, but the ions are still going to come out because it wouldn't work otherwise. It's the release of these charged particle ions that makes it kill bacteria. And it's sold in the clothing advertised as stink-free or smells fresher longer, or it kills the bad odors, things like that. And it does work. It does kill the bacteria that cause bad odors. So in camping clothing or workout clothing or your yoga clothing, it will work like that, but it will still release ions. So there's some challenges to engineering nanomaterials safely, which is really the overall goal, to test them in advance. That's the whole point of Greenscreen, is to know early in a research and development stage how hazardous your material might be, and then see if you can either modify it or ditch it if it's too toxic. So there's a group in Oregon that I'm sure you're aware of, a green chemistry group that's working to develop benign nanomaterials, and they're considering things like size and shape that can actually change the toxicity. So how could you design during the engineering phase nanomaterials that are safer? There's some principles that for regulating nanotechnologies that many groups have signed on to, many environmental and public health groups have signed on to, that are advocating a precautionary approach with nano-specific regulations. I'm not actually sure we need nano-specific regulations. I think effective chemical regulations that capture new chemicals effectively would work, but they do have to consider the small size. You do have to consider the nano size. You can't consider silver and nanosilver to be the same, and you can't consider carbon nanotubes and carbon to be the same. They have different toxicities. Also consider the health and safety of the public and workers. There should be protection of the environment. There should be transparency about when and where it's being used and what the hazards are. Public participation in the conversation, developing oversight, and also consider the broader impacts of these nanomaterials. These groups are talking about things like if there's a lot of electronic waste, for example, a lot of your new electronics, all of your new electronics are going to incorporate a lot of nanomaterials, a lot of metals. Those are going to get stripped out and cleaned out in developing countries a lot, where they're recycled and pulled out and reused, and that could cause a huge health impact to workers in those countries. So is this a situation where the global north is getting the advantage of new medical treatments or new and improved consumer products, but the global south is taking all the risk and hazard by doing a lot of the manufacturing and then recycling at the end of life? And also liability. Manufacturers should have to take responsibility. So in comparison, I think the project was a success. You can use Greenscreen to screen these. EPA could have used it when they screened the ones that it's approved. You can use existing toxicology and identify data gaps, which could drive research, which is really important. The other thing about the data gaps is, depending on how the material is used, you may not want to fill all the data gaps. It may be something that's going to have an aquatic exposure, but it won't have something else. And so you could decide where you want to fill the data gaps and where you don't. So you could actually drive targeted research, and that would be very helpful. You could also ensure nanomaterials are screened before they're introduced into food and other products, and it would identify, again, where you need more testing, depending on the use of it. We could require it. We could all, as advocacy organizations, require that things be screened in a validated and public and transparent screen done by an independent contractor, like a Greenscreen, and that those results be made public. There are several different companies that have nano policies now. Nike is one of them. They had one in 2007. They updated it in 2011. And they basically say that they're restricting their use of nanomaterials to ensure that any potentially negative impacts to consumers and the environment associated with the use of nanomaterials are minimized, if not eliminated. It's not clear how they do that, but they have a policy. GlaxoSmithKline, which makes pharmaceuticals, also has a policy. Their policy is a bit more vague. It defines nanomaterials, and it summarizes different positions on nanomaterials, if there are any, around the globe. And it confirms that they are using nanotitanium dioxide in sunscreens, and they are using some nanomaterials in their vaccines. The medical delivery potential for nanomaterials is very high because of its very small size. You can actually target it right to cells or tissues that you really want to, and you can actually coat, for example, the nanomaterial with some kind of a tag that would take it right to the cells that you want to deliver. So you could have very personalized medicine with these. McDonald's and Kraft also have nano policies. They state that they don't use nanomaterials in their food packaging, or in the toys that come with their food. But they are researching it for future possibilities, so they're open to it. Adidas, these companies don't have policies, but they have disclosed with some transparency that they're using nanomaterials, which is a step forward. Adidas says it only uses it in very single cases. Its golf shoes are coated with a waterproof layer that's nano. Marks & Spencer says it would only use nanotechnology where there's a proven customer benefit and where they know it's safe to use. Again, I'm not sure how people determine that. And they say at the moment it's not in any food or drink products, and it is used in some beauty products. There are a lot of nanomaterials in a lot of cosmetic products, actually. Cosmetics is very poorly regulated. H&M is a clothing store. They're a European chain, but they're also here. They say they don't use nanosilver in their clothing, but they do use some nanomaterials in cosmetic products. Sorry. And IKEA says that it's remaining neutral and flexible about it, but not using it now. So I want to thank our funders, the CS Fund, for supporting the Coming Clean Collaborative for this project, and to NSF International for conducting these screens, and to remind you that they're draft at this point and they may be altered before being finalized. So any questions are welcome now.
Oh, we have a ton of questions. So.
Oh, okay.
I will start reading them, and we will do our best to answer them, and thank everybody for their patience. So the first question is, why did the team select Greenscreen instead of US EPA's DFE master criteria, especially if EPA had existing data to review and approve nanosilver?
So we chose the Greenscreen because it's used by so many companies, and it is used by Design for the Environment. So it is consistent with what they do. It's also consistent with some European approaches, so that's why we went with it. In addition, we had someone who was working with me who is from Greenscreen, so I had that direct advantage, and that was really important, so that Lauren Hine, my co-author, and I could work together through this process. And I could not have done it without someone that knew Greenscreen intimately. So that's why we did that.
Thank you. For nanomaterials, are there standard production methods, or would processing be a variable that might affect nano characteristics?
Processing does affect it. It's a very good question, and in fact, there's a huge amount of toxic chemicals that are used in making nanomaterials, depending on how they're made. For instance, nanosilver can be made by taking rods of silver and just basically putting them in sort of a washing machine type thing and bashing them around with a huge amount of solvents. So there's a huge amount of toxic solvents and other chemicals that are used to create nanomaterials, and that can be a large part of their toxicity.
The person who asked this has left, but I'm going to ask it anyway because I think it's interesting. He says, is PBT appropriate for inorganic metals?
I think, so that's a good question. I mean, I think it is, because I think it's characteristics that we want to know, but also the truth is they're almost always going to be, I mean, I think maybe always, they're going to be very, very persistent. And they may or may not be bioaccumulative, but they're probably going to be bioavailable. In other words, they're going to be able to penetrate through tissues easily because of their small size.
And also, this person has left, but again, I think it's a really interesting question. How does the absence of important data affect hazard ratings?
So that's a good question. The answer is that they go on what they have, and I'm putting up the screen here. This is a bit complicated, and actually this is part of why this is draft right now, is because we're going back and forth with the contractors right now. You might notice that some of these are italics. So italics means that there's less confidence in that data for whatever reason, but they consider that separately. So they score it based on the data they have, and then the data gaps are evident for viewing. An italics means they scored it, but they're less confident in that data. And so that's sort of where we're having some back and forth with the contractors right now about some of these different scores and whether the data, we're confident enough in it to score it at all, and a few things like that. So, but those are good questions, and I would say that there are discussion points in there in the answer.
I also want to assure everybody that these questions are being read in the order in which they were received, so we're going to make an effort to get to everybody's, but that's how we're approaching the questions, okay? So what about inhalation of fume silver? Why did the Greenscreen not identify that as a hazard?
Inhalation, that's a good question. As opposed to, well, so they're reading across, you have oral, dermal, and inhalation, but there's got to be an endpoint. So inhalation itself is not a hazard endpoint, it's a route of exposure, but there's got to be some hazard associated with that in order to fill in that block. So you can see for the nanosilver that it was very high. There was a red for inhalation where you had acute toxicity as an endpoint, and that was based on rodent studies. It was high, which is the red for repeated. So there was a concern for inhalation is the answer, but it was in the endpoints where there was data.
If ionic silver is the toxic species and ion generation increases with solubility, what low solubility silver compound caused the high ecotox seen in the first grouping?
So that was because it still releases ions. So it's not as much. So that was the answer is that was like a silver nitrate, for example, some kind of a soluble silver, like a low solubility silver. And so it was. I think we used silver nitrate and for that one, but wherever there was data, we looked at where there was data, but that kind will still release ions, and those ions are still very, very highly toxic to aquatic organisms. And remember that this isn't a risk assessment. So it would take a lot more of that kind of silver, presumably, to cause the toxic endpoint than it would for nanosilver, but this Greenscreen doesn't consider dose. It doesn't consider, it's not a risk calculation, it just considers purely hazard.
Isn't persistence to be expected for inorganics like silver? I think you've sort of addressed this already. And isn't clean production action working on different ways to handle inorganics within the Greenscreen?
So, I mean, the answer is yes, persistence is expected, and I don't know what clean production action is doing to handle it differently. If they are doing something, I don't know about it, which is interesting because I've been working through this so intimately with my co-author, Lauren, and she didn't mention that they were working on something, so I haven't heard. I would have thought if they were doing something, I would have heard about it.
Okay. How much better characterized would you say the acute mammalian toxicity endpoint was for the nanoscale metallic silver versus dispersed non-nano silver?
Right. I would say that there was, we had a bit more data on the nano version because of its registration, whereas the non-nano silver, the regular silver, there is some data, but most of the data on regular silver is aquatic tox. It was registered so long ago, and people aren't really studying the toxicity anymore. It's so known to be hazardous. Is that the answer to the question? Did they ask me the difference between silver and nano for mammalian?
They wanted to know how much better characterized the acute mammalian toxicity endpoint was for nanometallic silver versus dispersed silver, the non-nano.
The non-nano. Yeah, it was somewhat better. There was some more data because of the registration process, so they had to generate some studies. There wasn't a whole lot of data. I would say 2 or 3 studies. They were very compelling, actually. There were some rodent studies where they had fed it to rodents, oral doses, and pregnant rats, and then the pups. It not only went into, it passed the blood-brain barrier and got into the brains of the pups. It passed the testicular brain barrier, and it also got into all of the organs of the fetus. So that, as well as the adults, but I mean the fact that it passed blood-brain barrier and testicular in the fetus is very significant. In the adult rat, it also got into a number of the different organs, including the brain. And then in other studies, we know that it can damage cells. It has a redox reaction. It creates oxidative stress. Seems to be the primary mechanism of toxicity. And actually a new study just came out recently that wasn't a 2014 study that wasn't included in this Greenscreen that I think is very compelling. It was printed in the journal called Nanotoxicology, and it's called, they actually they found that when they orally treated pregnant adults, rats, then the pups, they followed them out over several months and found that even at, I think it was 3 or 4 months old, the pups who'd been treated fetally, that they had damaged sperm. They had poor sperm quality and sperm abnormalities, so based from fetal exposures. So I think it's a very compelling study, and that's part of the reason why this is draft right now, is we're trying to see how we can incorporate those into the reproductive and developmental tox categories.
This also sort of goes back to an earlier question, but I think it's a sort of extension of that. The Greenscreen includes a data gap analysis. Did you conduct it for the silver analyses you did as well? If not, why?
Yeah, I mean, we did, which is why all the data gap things are filled in where they are. So we did it the same way. We used the data that was available and did it the same way.
Okay. Somebody says, I was surprised to see that the Nano Action 2007 document did not call for labeling. Can you talk a bit about labeling requirements, if there are any?
There are not labeling requirements at this point anywhere, not in the U.S. and not in Europe. It's something that the companies have really, really fought. And actually, I kind of feel that labeling was in there. So I think it was under the transparency point, because it was definitely in there. And many groups are still arguing for it. It should be labeled. I mean, all they'd have to do is in an ingredient list, instead of saying titanium dioxide, they say nano-titanium dioxide. In order to do that, you have to agree on the definition of nano, and that's where there's a huge fight right now. Europe has sort of adopted a definition that if it's 100 nanometers or below in any one dimension, then it's nano, but there hasn't been a definition that's been hardwired throughout the agencies in the U.S. or across Europe, and so there's an argument about that right now. I think that that's an annoying detail. I think that if the material changes its properties at small scale, then it should be listed as its nanoscale, because those altered properties could also impose altered toxicity.
What do you think the likelihood, though, is of getting that labeling?
Labeling, I mean, labeling is something that the companies fight more than anything. In fact, when we think we're going to win on labeling, they pull it from the product. So on the other hand, the flip side of that is some things do label that they're nano, and that's because they think it's a market advantage. So there are some things that, you know, are advertising that they're nano, and the problem is without proper labeling laws, there are things that say they have nano in them that may not, and there are things that don't declare that they have nano that may.
What tests are being considered in the field to determine how weatherization affects nanoparticles released to the environment? How are they collected from the environment and analyzed?
Okay, none that I know about. I don't think there's any, so that's a really good question. There hasn't really been any long-term studies at all, no chronic studies, and that would fall under that. And as well, they can't be collected from the environment at this point. That's a huge problem, is that we really don't have any proper environmental monitoring tools for these. They can be detected and tracked in a lab where they're being made and used, or in a in a manufacturing facility, but we don't have the kind of monitoring we'd need to detect them in workplaces and stuff, or out into the environment and water. So this is a real problem because all of our environmental statutes rely on finding it. If you can't find it and you can't detect it, you can't enforce an environmental statute. So this is a real problem right now. Some of us would like to require that there be a monitoring device or method that's associated with a chemical registration, that you shouldn't be able to put a chemical onto market unless it can be tracked.
The next questions, I think, were an exchange between people who were, it was a question about the solubility of silver nitrate, and whether it's soluble, whether it's extremely soluble or not soluble.
The
conclusion was that it's soluble because all nitrates are soluble.
Yeah, it is somewhat soluble, and that's why we chose it. So it had a low solubility, and we thought that it was, our scientific team, we went through this actually, there's not a good answer for this. I mean, there wasn't we didn't have a really good parallel, but when we did the metallic nanosilver, we went back and we sort of sent this out for a sort of an informal peer review, and one of the outside scientists said, look, how is this different than my silver spoon? You guys have to do silver if you're going to really say nanosilver. If you're going to say anything interesting about the nanosilver, you're going to have to compare it to silver. So we said, all right, what kind of silver? And there isn't really a magic answer, but our scientific team, about half a dozen people, said that we could use this one because it was a low solubility, it's somewhat solubility. It seemed to mirror as close as it could the kind of places where the nanosilver would show up. What we were concerned about was getting into water in a life cycle.
Okay, great. If nanosilver is discharged in wastewater, presumably it would be either incinerated in a sewage plant or used in land application for fertilizer. Would nanosilver be destroyed by high temperature incineration?
No, nobody said that, so I have not heard that. What I've heard is that it actually, in a wastewater treatment plant, it will settle down with the solids, and it will stay in the solids portion of the waste, and so it may end up as sludge on a field, for example. And again, as I mentioned in my talk, that the wastewater treatment folks are concerned that it would actually kill beneficial bacteria and prevent treatment of the sludge. But it would have to be in high volume for that. So that their concern is if the uses expand.
So somebody else comments that silver nitrate is very soluble. Silver chloride is an example of a low solubility salt. I'm just
So we did consider, we did consider silver chloride as well, and I would actually have to go back to my notes, but
could you please provide online all the research papers you have mentioned, and if you have a a reference list or something that you could send to us, we would be happy to post it if it's in shape, or if you're able to provide that to us, we would be happy to post that. I don't know if you have something like that available for the study.
Yeah, so I can't right now, but our hope is that we can get this in form to publish, and at that point, then we have the supporting documents, and we would make all that available. So, yeah, that's our goal for sure. But they're all, I was going to say, they're all public studies, or else they are the studies in the docket for the AGS 20 for the silver silica one. Composite. No, I can't find fast enough oh yeah, we did use silver nitrate, sorry. And I'm looking at my notes here. It says that this Greenscreen did not use silver nitrate as an analog to fill data gaps for the nanosilver, but EPA did. And
I'm going to have to draw this to a close because we're at 2 minutes to 3. I want to thank Jen for this fantastic talk. I think people were really into it, as you can tell by the number of questions that we had.